my Chitku people, I welcome you all to second PU midterm revision for biology, and today I have chosen the two most important chapters, that is molecular basis of inheritance and principles of inheritance and variation. And over here in this particular video, we will be solving certain very important questions which have come in the previous years, and also I have took certain questions from the model question paper because we never know, those questions could be repeated as well. Yeah, and now let's see the question paper pattern. So children, your paper is divided into 6 parts. Okay, A has 2 parts, D has 2 parts, rest all of them are single. So A1 has MCQ questions. How many of them? 15 of them. If you attempt them all correctly, you get 15 marks. Okay. Part A2 has fill in the blanks, 5 questions. All of them, if you attempt correctly, 5 marks. Part B has short answers with 2 marks. Okay, 7 questions are there, but you have to attempt only 5 among them. So 5 into 2, 10. Now part C has short answers again with 3 marks. So 7 questions will be there. You again have to attempt only 5 of them. So 5 into 3, 15 marks. Part D, the first part of D will be having long answer, 5 marker question. 7 questions will be there, out of which you just have to attempt 4. Please don't go on attempting all the 7 questions, my dear children. Okay, it will waste a lot of your time. So please make sure you read the instructions really carefully before doing that. So basically 4 into 5, 20 children, 20 marks. And the last part also has 5 marker question. 3 questions will be there. Out of that, you just have to attempt 1. So basically 1 into 5 is 5 marks. Total 70 marks paper. Fine. Now, and you are scoring 70, right? So yes, the weightage, if you talk about both the chapters, principles of inheritance and variation has 13, over there it also has 13, so it's equal weightage, high tie that you see over here. And the number of questions or the type of question that you can expect from the blueprint that they have given, 2 5 markers from this chapter, 2 5 markers from that chapter as well, and 1 2 marker there. So 10 plus 2, 12, and 1 MCQ might be there. Similarly, 1 MCQ might be there as well. Works? Now let's get into the questions. So my type of teaching is where I'll be explaining you certain things along with solving the questions. Okay, not just the question, we will be explaining a bit more. Fine, so that you feel that okay, we are covering a lot and it will be helpful for you to understand things. Now select the incorrect match from the following human karyotype characteristics. Okay, so the first one is 45 plus XS broad palm with characteristics palm crease. Children, this feature is of a disease which is known as, condition which is known as Down syndrome. Okay, and what happens in Down syndrome? What happens in Down syndrome? So basically, how many number of chromosomes do we have? All of us, we have 46 number of chromosome in total. Okay, in total we have 46 number of chromosome, out of which 44 of them are autosomes. That is, it does not have anything to do with the sex of the organism. It just codes for the proteins and everything. And then we have 2 more chromosomes, okay, either XX or XY. Both are sex chromosomes. Both are sex chromosomes. So in total, how many of them? 44 plus 1, 2 or 1, 2. Basically 46. But here there is a twist. In the case of Down syndrome, the 21st chromosome has trisomy. As in, we get 1 pair from father, 1 pair from mother. Basically here we are having 3 of them. Okay, how many? 1, 2, 3. So instead of having 44 number of chromosome in the case of autosomes, they will be having 1 extra, so it will be 45. It will be 45 plus XY, or it can be XX. So in this case, it is written 45 plus XX. So this is a female who has Down syndrome. Okay, so this is correct. It does not have any problem. Okay. Now next is 44 plus XXY, overall feminine development. Now this thing 44 plus XXY is basically a case where you have normal 44 chromosomes, agree, but the sex chromosome, if you see, it was XY, sorry, but you have an additional X over here which makes it XXY. In this condition, first of all, what is this condition known as? It is known as Klinefelter syndrome. This is known as Klinefelter syndrome, where it's not overall feminine development. No children, it is actually overall masculine development, but there will be certain feminine character, as in there will be formation of breast and everything. Okay, gynecomastia is also there. Fine, this is the case of Klinefelter syndrome. So this is wrong. This statement that they have written is wrong. So this is the incorrect match. This is your answer. I'm writing as answer, but I have to explain this also. Okay. Next thing is 44XO, sterile females as ovaries are rudimentary. Over here, what is happening? So basically, they are male only. They are more male with feminine characters. Similarly, over here in this case, as you can see, 44 is there. So it's all cool. 44 autosomes are there, so autosomes are all good. But you see XO, what do you mean by XO? So that means only one chromosome is there, sex chromosome is there. Next sex chromosome is not there. Total how many chromosomes are there? Just 45 chromosomes are there. One chromosome is altogether missing, children. So over here there is only X. So similarly, this particular person will be having a syndrome which is known as Turner syndrome. This is known as Turner's syndrome, and over here it will be a female. But that female will be sterile, children. That female will be sterile. Okay. Now 44 plus XY is normal male. S, you know, autosomes 44 are there. X and Y 1 and 2, 46 in total. This is a male because X and Y is there, so it has to be a male. If X and X was there, it has to be a female. Got it? So this is your answer. I hope you understood why is that the answer. Works? Let's go ahead. Yes. Question number 2. The type of bond represented by the dotted line in the schematic polynucleotide chain is. So this is a polynucleotide chain where S represents sugar, children. And your sugar, it looks like this. Okay. This is the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon, this is the fifth carbon, and on the fifth carbon you have the phosphate group. Okay. On the first carbon you have the nitrogen base attached. Fine. And usually it is not made, but I'll make it. So on the third carbon you have OH. Okay, usually it is not made. And now if I make another sugar molecule over here, okay, again 1, 2, 3, 4, 5. On the fifth carbon, what do I have, children? I have the phosphate group. This phosphate is going to attach to that, and that is how things are being made in the case of what? DNA. So DNA strand goes like this. Okay, DNA strand goes like this. Now here only I'll explain you a thing. Over here you see above this there is no sugar, so this phosphate is free. So I call this as 5' phosphate end, or basically I just call it 5' end. Okay, so if this is a strand, this is the 5' end because it has the phosphate group as a free group. Over here if I see the 3OH is not attaching to the next one, so over here I can say it's a 3' end group that I can find over here. So this end will be 3' end, children. Okay, this is how DNA works. Now this is deoxyribose. This sugar that you see over here, it is deoxyribose. So in the case of DNA, you have deoxyribose. In the case of RNA, you have ribose sugar. What is the difference between deoxyribose and ribose? Oh, basically in the case of ribose sugar, in the case of RNA, it will be having an OH group over here. But in the case of DNA, there is no OH group. There is no OH group. Fine. Now the question is, what is the bond that is being made between the sugar and the base? This is the sugar and this is the base. What is the bond that is being made over here? This bond you call it as glycosidic bond, children. What do you call it? You call this as glycosidic bond. Okay. Now next question, if it comes, what is the bond between the sugar and the phosphate? This is an ester bond. This is a phosphoester bond, phosphoester bond. Okay, so here also there is OH group that is attaching to the P. Here also there is OH group. Here the OH group is not been mentioned, so which is why it is like that. Okay, now here also this is also phosphoester bond. If I say together the name of the bonds, if I say together both of them 1 and 2, I can together call them phosphodiester bonds. Phosphodiester bond. Cool, children. Now in between a nitrogen base and another nitrogen base that will be coming around, this will be hydrogen bond. This will be an hydrogen bond. Okay, so over here the correct answer will be peptide. Peptide comes between amino acid. Okay, peptide, amino acid 1, amino acid 2. This bond that you see, that is peptide bond. This bond that we see over here is N-glycosidic linkage, so you give a tick on that. Works? Yeah. Next thing, you know that there are 20 different types of naturally occurring amino acids. We know that, right? Now, a combination of 3 such bases code for specific amino acid. So who said that 3 amino acids are coming together to form the the codon and everything? Basically, how did we find out is that if 20 amino acids are there, and in total how many nitrogen bases do we have? A, T, G, C, that is equals to 4. We have 4. So if the 4 repeats twice, it is 16, but still it is not close to 20. But if we say 4 raised to the power 3, it's 64. It's more than 20, but at least we need these many. We need these many set of codons to code for. Now some codons among them. So I'm telling there are 64 codons like A, U, G, 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 A. Similarly, how many of them? 64. And what are the number of codons over here? 3 of them, they come together to give like this. So you have 64 of them. I'm not saying 64 of them is coding for individual amino acids. No. Few of them might be same. Few of them might be coding for the same amino acid, but at least all of them are coding for amino acid. Only 3 among them are coding for stop codons. They do not have any amino acids. They are coding for stop codons. So how did we find out? Basically 4 bases are there. So 4 is to the power of whatever thing is there, that will be minimum near to this number. We will get to know that these many codons are there. Okay, and these many nitrogen bases have to come together to form a codon. That is the thing. So over here, if I say the question is asking a combination of, if instead there are 96 different amino acids. Now situation is like this, where there are 96 different amino acids and the different bases that you see are 12. So instead of 4, here how many of them are there? 12. Okay, so if I think basically if they come in a pair, okay, so then in that case it will be, okay, first thing, if they are coming alone, if I am saying that basically there's 12 bases, they are coming together in different combination. In that cases I will be getting only 12 amino acids. In that case I will be getting only 12 amino acids. But how many of them do I need? 96. So obviously this is not possible. So what do? I change this number. I try for 2. Okay, so 12 times 2, basically over here I get 144. 96 is the number of amino acid. If I have 144 codons, I can obviously code for 96 amino acids. Some of them might not code for amino acid. They might be stop codons. Some of them might code for the same amino acid. So many codons could code for example, this XXY, XXX might be coding for the same amino acid, right? But they can do all of this thing, right? That is possible. So this is the closest number that I can get. So with this information, I get to know that the number of combination that should be there, it should be at least 2. Over here, it was at least 3. So here, it should be 2, and with this, we get this as the answer. Fine? Works. I know this is a little tricky, but then we have to study. Going ahead, assertion and reason question. Linked genes do not show dihybrid F2 ratio, that is 9:3:3:1. What do you mean by linked genes? So basically, this is there, this is a chromosome, and on the chromosome I say this is a gene, this is another gene for another character. Okay? There are 2 genes. Now, basically, whenever I am transferring this to another organism, the offspring, basically, when a copy is being made, this will always remain like this only because they are very much close to each other. Okay, they will be remaining very much close to each other. Okay, so for suppose this is for height, that is tall, and this is for the color, that is white. Always the person who will get this thing will be tall and white, because it's very closely linked. The genes are very close. But on the other hand, if on one chromosome, this was the case, on one chromosome I had the thing for tall, and on other chromosome I have the thing for color. Okay, in this case they are not linked. That means this chromosome obviously can be copied and can be passed down. Again I have an option, either this chromosome or another small chromosome or another chromosome could be passed on. So it's not always necessary that the capital W goes with capital T. This time this chromosome and this chromosome can also go into the gamete, right? It's independently assorting. It is independently assorting. It's not saying that I'll go with you only. That's not possible. But over here, does it have an option? No, because these genes are closely linked, but over here they are not closely linked. Okay, so basically if this wants to go from, if there is a pair and over here I have small t and capital T, over here capital W and small w, this is for color and this is for height. If this wants to go with this will be the condition, small w. Okay, if this wants to go with this, then this will be the condition, capital T, capital W. Anything can happen. That is what you call as law of independent assortment. So in this feature, that is dihybrid thing, we get 9:3:3:1 ratio because we have that many features. They are all allocating together, I mean differently. But on the other hand, if you think that this was the case, then probably you won't get these many different types of genotypes there. You won't get these many different types of phenotypes there. Always, all the plants will be tall and white. In this case, it will be always tall. If this is tall, it has to be white. You won't get tall and dark. Never. You won't get that because these are very closely associated. Okay. I hope I was able to explain you this. I hope it was explainable. Now, so you understand one thing, that if linkage is close, that means you would not find no much amount of variation will be found. Okay, linkage is not close, then the opposite of it, there will be variations that will be seen. Okay, now linked genes do not undergo independent assortment. Linked genes, as I say, do they undergo independent assortment? Independently assorting means basically this is independently getting assorted from this. That is possible here? No, it is not possible over here. They are closely linked, so it won't be getting independently assorted. So both the statements are correct. Linked genes do not show dihybrid this thing. Linked genes do not undergo independent assortment. Okay, so and this reason is explaining that thing. Yeah, linked genes do not show this particular thing because they are not undergoing independent assortment. So assertion is correct, reason is correct, and the reason is explaining the assertion as well. Okay, so both assertion and reason are true, and the reason is the correct explanation of assertion. I hope it is clear. Now if it is not clear, because I feel that I didn't do justice to the question, so I can try it once more. Listen to me very carefully. Okay, in the case of round and yellow seeds, you studied this. Okay, capital R was for round and this was for wrinkled, and then there was another chromosome, this was for yellow and this was for green, right? So over here the chromosomes which are copying and going in the gametes, you know that only one, from a pair of chromosome only one can go down to gamete. So in the gamete, one type of gamete could be capital R, capital Y, another type of gamete could be capital R, small y. This can go with this can go with this also. Similarly over here also, the gametes can be small r, small y, or it can be small r and capital Y, because they are showing independent assortment. Okay, the genes that you see over here, they are not closely linked. So these many gametes are possible, and the gametes they fuse with another gametes given out by different parent, obviously they will have different type of variation. But in this case, when I am saying it is slightly linked, so it can be linked like this, where capital R is over here and capital Y is over here. Small r is over here and small y is over here. So obviously when the gametes are formed, from one can go, from here also one can go. So it always, whatever gametes will be forming, it will be capital R, capital Y, and this will be small r, small y. Every time, because this is linked, they are closely linked. So you did not see an option there. Over here you saw two options. Over here you did not see an option there because they are closely linked. Now, if this is happening, if these set of gametes are formed, that's when we say 9:3:3:1 thing is being formed. That is, 9 of them will be round and yellow, that is the parent feature, okay, round and yellow. 1 of them will be wrinkled and green. 1 of them will be round and green. That is a different feature that we didn't see until now in the parental generation. So over here you won't see that. Okay. Now I hope it's very much clear to you. Done. Let's go to the next question. Listed below are some amino acids and their corresponding mRNA codons. So these are the mRNA codons, phenylalanine, lysine, this and that. Select the DNA sequence that could code for the polypeptide sequence. So alanine, arginine, you have to find the DNA sequence, children. Okay, so there is DNA. On the basis of DNA, you get the RNA, and this is what? mRNA. So these are the mRNA codons that you see over here. What is the mRNA codon? UU. First thing you have to find about alanine, because alanine is the first thing over here. So alanine, let's say, is GCA. If in alanine the mRNA is GCA, then in DNA, complementary to G, what do you have? C. Okay, complementary to C, what do you have? G. Complementary to A, what do you have, children? In the case of DNA, it's a DNA, not an RNA. So in the case of DNA, it will be T. CGT has come, the first thing. Okay, CGT is the first thing. Going ahead, what do we have? Arginine. Arginine over here is CGA. Arginine over here, it is CGA. Right? Now we have to make a complementary DNA, that C is going to code with G, G is going to code with C, and A is going to again code with T. Okay, this is GCT. I hope you know that A is friends with T and G is friends with C. How many bonds are there in between G and C? 3 bonds. I always tell, I have already told in the NEET class also, G and C look close, very similar. So we say people who are very close, they start to look similar also. So they have 3 bonds that makes them very close, and A and T have just 2 bonds. Okay. Now in the case of RNA, it's replaced, the T is replaced by U. If it was RNA, it is replaced by U. Okay. So just understand, instead of T, if U is mentioned, just understand it's for thymine. So you have to find the opposite, and that in A. Now over here, we'll come to that only. We'll come to that. What do you have? Lysine. Lysine is AAG. AAG. Right? So over here, what will you have? Instead of A, it will be T. Here also it will be T, and instead of G, it will be C. Now phenylalanine is basically UUU. Phenylalanine is, children, UUU. Okay? Now UUU, then here it should be A. You know, right? U is going for A. So it will be A. Tell me, which is the codon? So CGT, GCT, TTC and AA. A is the correct answer. Fine. If they are asking about anticodons, you have to tell the opposite of this. Instead of T, it will be U, if it is for anticodons. Okay. What do you mean by anticodons? Basically, this mRNA, for example, UUU is read by an anticodon, that is tRNA has the anticodon. Okay, tRNA will be having an amino acid onto it, and on this end it has an anticodon which codes for this amino acid. So basically UU, then it will be A over here, and it will be having a particular amino acid. UUU stands for phenylalanine. So this will be phenylalanine. Cool? Works? Going ahead. Match the items in column 1 with those in column 2 and select the correctly matched option for those given below. Okay, so we have to see column 1 and column 2. So I have made over here, okay, basic things we should be knowing. So Mendelian monohybrid, okay, Mendelian monohybrid, you know it's 3:1, that is F2. How did we get it? I'll tell you. So capital T, small t, this was done by Mendel. Okay, this is first case. Mendel monohybrid, as in you're talking about one character, and as of now I'm talking about just one character, that is height. So with the gametes that is formed is capital T, or these are the number of chromosomes. Okay, so either this chromosome or this chromosome can come in. Similarly, either this chromosome or this chromosome can come in, right? So these are the gametes. Now F1 generation will be having this. F1 generation, if I fuse this with this, I get capital T, small t. This gamete can fuse with this gamete, right? Similarly, this gamete can fuse with this gamete, again capital T, small t. This with this, again capital T, small t. This with this, again capital T, small t. All of them are same. All of them are same in F1. Now F2 basically means I take, these are the parents, capital T, small t, capital T, small t. Now these are the parents. What will be the gametes, children? Either this chromosome can come or this chromosome can come. This chromosome can come or this chromosome can come. Now if I fuse, I get this and this, that is capital T, capital T. I get this and this, that is capital T, small t. I get this and this, that is capital T, capital T. I get this and this, that is capital T, small t. Okay. So you see two capital T are there. Oh, did I mess up? Wait. Just a second, okay. Fuse this with this, I get capital T small t. When I fuse with this, I get capital T small t. And if I fuse this with this, I get small t small t. Yeah. So over here I see capital T is 1. Capital T small t, I see 2 of them are there. And small t, 1 of them are there. Okay. If I see this over here, this capital T is there, both of them are capital, basically it's tall. If here capital T small t is there, capital T will overshadow this effects, so it will also be tall. Over here this will also overshadow, so 3 of them will be tall and 1 of them will be dwarf children. So the ratio is what? The ratio that I get is 3:1. This is what the phenotypic ratio. Phenotypic ratio. And what do you see this as? This is the genotypic ratio. How does the genetic makeup look? So genetically they are like this, 1:2:1. This is the phenotypic ratio. This is the FF2. So Mendelian monohybrid with this thing. Okay, Mendelian monohybrid with this thing. Incomplete dominance. In the case of incomplete, okay, before that, test cross monohybrid. Test cross basically means over here I have a parent like this. Okay, I am saying there's a tall parent. Now I don't know whether it's tall like this or it's tall like this, because in this case also it will this is dominant one, so this will overshadow the recessive one, right? So for that I do a test cross. And in test cross what is done? Basically I take the parent plant, for example I am taking it first, and I am fusing with a recessive, pure recessive parent. So pure recessive parent I have taken. Now when I cross them, when I cross this, I get capital T small t, capital T small t, capital T small t, capital T small t. All of them will be tall, all of them will be dwarf. But if the genotype is actually like this, the genotype, it was not like this, it is like this, then the things are quite different. Then over here, when I fuse, okay, capital T, small t, small t, small t. When I fuse this with this, I get capital T small t. When I fuse this with this, I get capital T small t. When I fuse this with this, I get small t small t. When I fuse this with this, I get small t small t. So I see 2 of them are dwarf because small t recessive condition is there, and 2 of them are tall. So 2:2. If I cancel it out, 1:1. So for test cross, okay, now I understand by this thing that if it 1:1 ratio that is coming in, then probably this is not homozygous tall. Homozygous tall means when both of them are same. It is heterozygous tall because one is different and another one is different. Okay, so this is for test cross. Okay, now for Mendelian dihybrid we know it is 9:3:3:1, and for incomplete dominance children it is 1:2:1 in F2 generation. Okay, in incomplete dominance you have 1:2:1. If you want me to explain, I can explain it to you right away. Let me explain it over here. And in the case of incomplete dominance, what happens children, there is what was it? Snapdragon had pink flowers, capital R, and red flowers and red flowers and then white flowers. So this is for white, this is for red. Okay, this is pure red and pure white. Now when I try to breed, I get capital R. This is the gametes, small r. Please write gametes and then explain all this thing. Okay. Now when I fuse this with this, I get capital R. Same thing. So all of them, when I saw, okay, genotypically they are all same, phenotypically they are all pink. This was red, this was white, but the progeny are all pink. Okay, now I do the FT, selfing with them. So there is capital R, small r, capital R, small r. The gametes are capital R or small r, capital R or small r. When I fuse this with this, I get capital R. When I fuse this with this, I get capital R, small r. When I fuse this with this, I get capital R, small r. When I fuse this with this, I get small r. So over here, 1:2:1, 1:2:1 is what the genotypic ratio. Genotypically this is only one type. Genotypically this is also, both of them are same, so 2. And then genotypically this is also rare, this is only one type. Phenotypically also, if I see phenotype as in how do they look? Phenotype, how do they look? How do the physical appearance? Phenotype is for physical appearance. So capital R is basically red. Capital R, small r, as you saw, it is pink. and small r, it will be white again. It will be white. So two of them, one of them are red, two of them are pink, and one of them is white. Again 1:2:1. So over here for incomplete dominance. So why do I say incomplete dominance? Over here, if it had been completely dominant over small r, then this would also have been red. But this is not completely dominant, this is incompletely dominant. Okay, that's why it's pink in color. So in F2 generation, this is 1:2:1 for that also. Okay, works. This ratio, if I try to bring in now, it will be too much time taking, but I will show you, okay, in the same lecture. Okay, in a transcription unit, the terminator is located towards, what do you mean by terminator? So my dear children, over here things looks like this. Okay, when I say transcription happens, you know there is two helix of DNA, two strands of DNA. There is two strands of DNA which are coiled among themselves, right? So over here, the strand which is in 3' to 5' direction, okay, this strand will be known as the template strand. What do you call this? A template strand. The other strand opposite to it is coding strand. And in what way? Transcription means what? Transcription means the formation of mRNA from the DNA. Okay, from the DNA, mRNA is formed. So mRNA is formed in 5' to 3' direction. That means from here it will start. From here it will start forming. So this is 3' end. So this is 5' end. So from here it will go to that end. Okay, so what is the direction of mRNA formation? 5' to 3' end, children. Fine. Now, who does it? RNA polymerase. RNA polymerase looks for a site, that is promoter site, and binds on it. So this is the promoter. Okay. Then this is the gene which has to be transcribed. This whole region that you see is a region where it has to be transcribed. When you talk about terminator, terminator is over here towards the end. Towards the end. Okay, that means termination will be taking place over here. Now, if you are asked about the location of the terminator or the promoter, you do not look for the template strand. You do not look for the template strand. You give reference to coding strand, because coding strand anyways does not have any work until now. So to not make it feel bad, we give reference to coding strand. So we say promoter is located towards the 5' end of the coding strand, or we say a terminator is located towards the 3' end of the coding strand. Rather than saying 5' end of the template strand, which is actually getting transcribed, we are saying 3' end of the coding strand where the terminator is located, and 5' end of the coding strand where the promoter is located. So over here you are asked about the terminator. Okay, you are asked about the terminator. So terminator is located at the 3' end of the coding strand. This is given in your NCERT, this piece of information. Okay, works? Can we go ahead? Next question. In the double helical structure of DNA molecule, the strands are, when we say double helical structure, okay, I told you how 5' end is formed and how 3' end is formed. So when there is no one to bind with the phosphate, you say 5' end is free, and when there is no one to bond with the OH of 3, okay, you say this is free. So I will show you yet again once more. So this is the sugar. What is this sugar, my dear children? Can you tell me in the comments? What is this sugar? This is the 5th carbon, and over here you have the phosphate group. On the 3rd carbon you have the OH. Now suppose this particular DNA strand, just like this, you have so many DNA strands, and at the end 3 OH group is free. Usually this binds with the next phosphate of the next sugar. But if this is not happening, you say 3' end is free, because it is like this, it is not binding to phosphate. Similarly over here also 5' is free. Okay. Now if one helix is like this, then the other helix, children, will be opposite of this. So here it will be 3' and here it will be 5', and I will give you an example of how does it actually look. Okay. So this is the sugar, this is the nitrogen base, this is the phosphate group. Okay. Over here this is the 1st, 2nd, 3rd, 4th and 5th. Okay. Now how do you make it there? You have to make it ulta. So this will be NB. Now over here the sugar will go like this. Okay. This will be the 1st, 2nd, 3rd, 4th, and this will be the 5th with phosphate group. So here you see 3 OH group is free there, and here you see 5th phosphate is free. So that's why we say it's 5' end, and this is what? 3' end. Similarly, if you take ulta of this, you get 5 at the end, and here you get 3' at the end. So it will be what, children? Are they complementary to each other? Yes. Why do you say complementary? Because here if it's A, there it is T. Here if it is G, it will be C. So they are complementary to each other. Okay. A does not go and bind with C. T does not go and bind with G as well. Okay. So they are anti-parallel. Okay. They are anti-parallel. Ulta hai na? So we say anti-parallel and they are complementary. Works? Done? Going ahead. A woman with normal vision has a colorblind father. Let's do it. So most interesting things. I found these many questions in NEET also. A woman with normal vision has a colorblind father. So for a father to be colorblind, okay, color blindness is a recessive character, is a sex-linked recessive character. Okay, so I am trying to say that basically if it's X and X, okay, it's X-linked recessive character, then if there is one X chromosome that is affected, then this particular person will be just a carrier. This person is not colorblind. This is just a carrier because it's a recessive character. For recessive character to show up, both of them should be having this. If this was dominant character, then only one was needed, then it would have been anyway shown. But this is a recessive character. For recessive character, both of them should be having the problem. Okay. In this case, when I am saying father is, I would have fallen right now. Yeah. So if the father is colorblind, so father is basically X and Y, and this is what say a recessive trait over here. X will always have this case if the father is colorblind. There is no other option. Okay. A woman with normal vision has a colorblind father. She marries a man with normal vision. So if the father is colorblind, then the daughter will obviously be a carrier, at least if she has a normal vision. She has a normal vision, then from father she had got this, but from her mother probably she might have gotten normal X, which is why she has normal vision, as she is just a carrier, my children. She is just a carrier. Now this carrier does not know that she is a carrier. Now she marries what? A man with a normal vision. She is going to marry a man with normal vision. This guy is surely having normal vision. Why? Because man, if that man had C, then this man would not have normal vision. This man would have the color blindness, because only one X is needed, right? Now when I try to fuse them, okay, so the chromosome can be X, the gamete can have this, or the gamete can have from this end, it can be this. Now when I fuse them, this and this, when it comes together, it's XX. Okay, this and this, when it comes together, it is XXC. Over here, when this and this comes together, it is XY. Over here, when this and this comes together, it is XCY. Over here, you tell me who are the carriers, who are affected. This is totally normal female. Okay, if you see over here, this female is a carrier. Okay, so I can say out of 50% of female, this is 50% of female, this is 50% is male, right? Out of 50%, 25% female is a carrier. Okay, now if you talk about boys over here, this guy is normal, this guy is color blind. This percentage is color blind, because there is no other X to overshadow the effect of this, right? So this person is color blind and this is a male. So how much percent, children? 25, 25, 25, 25. This is 25% of the guys that the progenies will be produced who will be colorblind. 25% will be just the carriers. They are not the colorblind people. So the percentage chance of their progeny being colorblind is, children, 25%. Okay, got it? I find very interesting this questions to be very interesting when it comes to color blindness. If you know these things, it's very easy. Now let's solve the 10th question. Assertion is that RNA is unstable and can mutate at a faster rate. Okay, do you know that RNA is the first genetic material that was there? RNA was the first genetic material, and later on, when there were so many changes in the RNA had this ability to mutate really fast. It was very unstable. It is exactly true that they have said. Okay, why is that so? First thing, I told you the sugar in them, it was not like deoxyribose sugar. The sugar in them had OH group on the second carbon, and this OH group was very reactive. It randomly gotten reacted with lot of things, which is why the structure and everything changes very now and then. So it was like that. Okay, that is why it was considered to be unstable, and it was considered to be mutating at a very faster rate, children. So that is why a more stable form of genetic material was created, that was DNA, where certain changes were made. This thing was basically removed. OH group is not there in them, right? OH group is not found in them, and they were mostly double helical DNA. When you say it's double helical, this could have existed in single form also, single stranded form as well. Cool, works. Now over here, so RNA is unstable and can mutate at faster rate is exactly true. The presence of 2H group in every nucleotide of RNA makes it labile and easily degradable. Yes, children. This is the reason. Assertion and reason is true, and both of them are explaining each other. So basically both assertion and reason are true, and the reason is the correct explanation of assertion A. Works? Done? Can we go ahead? Okay. Now 37.2 recombinant Drosophila progeny obtained in the T.H. Morgan's dihybrid cross experiment with the phenotypes red eye color, normal body and white eye color, miniature body is due to. Okay. Basically, they are saying out of 100%, okay, out of 100%, if an amount that is 37% of the progeny are recombinant, that is not like the parents, okay, they are showing variations, they are recombinations been happening, and it's a huge number. Usually the percentage of recombination will be 1.3 also, 1.3%, which is 37%, okay, 37.2%. So this is kind of huge. So when I say that recombination is happening and I am finding different features onto it, okay, when does this happen? When the links are closely linked, or when the genes are closely linked, or when they are loosely linked? How do you understand? So basically I told you already, this is a chromosome, and if this chromosome, there is one character, another character closely linked at this point of time. What is recombination? Recombination means basically one part of this goes to that part, one part of this goes to this part. That's also how you get variation. But you think if this part is going all together, anyways you have the same character over here also. Whatever you do, this is like together only. Okay. So recombination, when I say closely linked genes are there, when I say closely linked genes are there, the recombination will be less. Recombination means basically this only. One part of this is going that side, one part of this is going this side, which is why I have variation. Okay. For example, look here. Okay. So capital T was here, capital W was here, small w was here, and small t was here. When I try to recombine, suppose this is getting cut and going that side. Okay, and this is getting cut and coming this side. So this time the new one will be having capital T over here and small w over here. So I got a new variety. Okay, now this particular chromosome can go to the gamete. So anyways, this is different from the parental variety. So in the parents only this thing happens. But this will only happen when there is a distance between the genes. So closely linked genes will show less amount of recombination. When you talk about loosely linked genes like this, they will show what? High amount of recombination. Okay, so loosely linked and shorter distance between. Okay, so it is because, so 37.2 recombinant, that means good amount of recombination has taken place. That means, are they closely linked or are they loosely linked? They will be loosely linked. That is why recombination is happening tremendously. Okay, so loosely linked and shorter distance between the genes. If they are loosely linked, then how can they have shorter distance between the genes? Okay, tightly linked and shorter distance, no. Loosely linked and longer distance between the genes. Tightly linked, and so basically it's loosely linked and it has longer distance between the genes. That's when this much amount of recombination can take place. Okay, children, got it? Now, the number of nucleotide pairs present in the DNA of the primary oocyte. Children, primary oocyte, you know, you're talking about the female gamete. And you also know one more thing, that female gamete, male gamete, both of them, they are haploid. That is, they have half the number of chromosomes, and when they come together, they fuse together, you get diploid zygote. you get what, children? You get diploid zygote. So haploid, if our normal ones have 6.6 into 10 to the power 9, then half number 1 will be this one, 3.3 into 10 to the power 9. Works? Cool? Going ahead. The initial transcript formed during transcription is known as. So the initial transcript that is formed, so we say that, okay, this is DNA, DNA is going to form mRNA, but is it like that? Does it directly form mRNA? No, children. There is an initial transcript that is being formed, which is not known as mRNA. Instead, this thing that is being formed will be known as hnRNA, that is heterogeneous nuclear RNA. Okay, and what is different from this and this? Basically, hnRNA will be having areas which will be actually coding for genes and will be having areas which would not be coding for genes. So, for suppose this is coding for gene, this is known as exon. This is also known as exon. The ones that does not code for anything, it is known as intron. Intron stands for intervening sequences. So, they're just there. They're not coding for any genes. They're not coding for any proteins. They're just there. So, when I cut this off, okay. This was in yellow, suppose, okay. When I cut this off, I get green thing. And this process is known as splicing. So when I am processing the hnRNA, what's the last thing that I am getting is known as the mRNA. The first thing that I am getting is known as hnRNA. So this is the answer, my dear children. And this is the fill in the times type. Fill in the blanks type of question. You will be given option and you have to fill. It's not like every question will have option. No, they'll give you 5 questions and then you will be having 5 options as well. You have to change and find. Works? Cool. Now, although hemophilia, this is 2-marker question, okay. Although hemophilia and sickle cell anemia are 2 blood-related Mendelian disorders, yet they differ in their pattern of inheritance. What happens in hemophilia? Queen Elizabeth had it. In the case of hemophilia, what happens is that the person who has it, okay, they're not able to clot the blood. For example, I get a wound. Basically, my blood will go on. There is no clotting mechanism in my body. My clotting mechanism is affected in this particular disease. Okay. Now, sickle cell anemia is when my normal normal red blood cell, which looks like a discoid, okay, it has a discoid shape. It looks something like this. Now, it is changed to sickle cell. Over here, obviously, why it is happening? Because our normal cells, our normal red blood cells, it has hemoglobin in it. It has what? Hemoglobin in it. Okay. Now, over here, in the case of sickle cell anemia, what happens is that hemoglobin molecules, the beta chain, the hemoglobin molecules have 4 chains. Okay. They have 4 chains. Alpha, beta, beta. Okay. These chains are globin chains, the protein chains, and all of them have the heme protein with them. All of them has heme protein with them. So what happens is that the beta globin chain of hemoglobin is affected in the case of sickle cell anemia, which is why the structure of hemoglobin is affected. Now, which is why the blood cell's shape turns out to be sickle. And will it be able to carry enough amount of oxygen? No, children. So the person who is having sickle cell anemia will die. Now, they are asking you, since both of them are blood disorders only, how can you differentiate them? Okay, how it is different? How is the pattern different? So, first thing first. Basically, if we talk about, okay, should I start from here? I'll start from here only. Okay, hemophilia, this is an X-linked recessive disorder, as in, for example, there is a male with XY and another female with XX, okay, and I say it's recessive. For recessive characters to be shown, they have to be together. For example, XH, both of them should be, both the chromosomes should be having this, then only it will be shown up, okay. In the case of this, when there are 2 chromosomes of the same type, both of them should be having this. If the condition is like this, then this will overshadow, the normal one will overshadow the effect of this, because it's a recessive thing, so it won't show up. Now, in this case, X and Y, if this is affected, obviously there is only one X, there is no one to overshadow the effect, so this will show up. This particular guy will be having hemophilia for sure, okay, will be having hemophilia for sure. Now, so males XY are more likely to express the disease because they have only one X chromosome, so there is no one to overshadow. Female XX are typically carriers and usually do not exhibit symptoms unless they inherit two faulty X chromosomes. So when that condition comes where they are exhibiting two X chromosomes which are affected, that's when the females will be affected. On the other hand, males, if they have at least one, they are going to be affected. Fine. Females, on the other hand, if they have one, then they are just carriers. Okay. So this is first thing that you have to write is this is an X-linked recessive disorder. On the other hand, if you talk about sickle cell anemia, it is autosomal recessive disorder. It does not have anything to do with the sex chromosome. It is autosome linked. That means autosomes, if for example any autosomes that you taken, okay, any autosome that you taken, in that case over there in them, what will happen? For example, HP. This is hemoglobin's one that is affected, so I am writing HP, okay. This is one chromosome, this is another chromosome. Over here, if both of them has the problem, if both of them, okay, wait, I have written too far, you won't be able to understand. So this is the chromosome, okay, which has the problem S. Now this is another chromosome, this is also S. In this condition, that disease will come. In this condition, that disease will be coming in. If both of them are getting two chromosomes or the same type. Now if this is the condition where one is normal, the other one is this, then this will overshadow the effect, and now what will happen? This person will be just a carrier. This person will be just a carrier. Okay. Now you tell me, can males have this more and females have this less? Is it like that? The probability, does it change? No, children. It is an autosomal disease. It does not have to do anything with the X-linked. Does not have anything to do with the sex chromosome. So basically it's about autosomes, and autosomes are basically a man can have two, a female can also have two of the same time, right? So basically if a man and a woman, both of them, basically if they are having this condition, that means they have sickle cell anemia. If they are having one, does not have. So this is autosomal recessive disorder. When this condition comes, that's when that particular disease occurs. In this condition, the other normal one overshadows the effect. So this is an autosomal recessive disorder, meaning the defective gene is located on one of the autosome. Okay, over here chromosome number 11, this is 1, okay, and both male and female are equally affected if they inherit two copies of the chain. Okay, that is one difference. The type of inheritance, the type of disease is different. Second type of difference that you need to find is carrier and expression patterns. Basically, females can be carriers without showing symptoms. In the case of hemophilia, I told you. Why? Because this chance is obviously there. Okay. Now, but if a male inherits a defective trait, then what will happen, children? Anyhow, that particular person will be having the disease. Okay, he will express the disease because there is no second X chromosome to mask the effect. Similarly over here, both the males and the females, both of them will be carrying the disease. Okay, so individual with one defective alleles are carriers, but both of them will be equally affected if they have this condition. So carrier and expression pattern is different over here. The inheritance pattern is different over here. That's how you can explain. And now I'll tell you one more thing which is given in your book regarding sickle cell anemia. That is, how does this occur, this particular globin chain, how it is mutated and what all happens. Basically, there is a codon that is GAG, and this GAG codes for glutamic acid. Okay. And this U is actually replaced by, this is a point mutation. Point mutation means only a single nucleotide is being changed. This is replaced by GUG, which is why now the codon is different, and this codon codes for valine. And this makes the whole change in the beta globin chain. Now it won't be normal. And this is the 6th codon, children. This is the 6th codon of that particular gene. Works? Okay? Now going ahead. Which sequence of bases transcribed from DNA are found both in hnRNA and mRNA? What are they trying to tell you? Or what are they trying to ask you? They are asking you, if this is hnRNA and this is mRNA, what are the sequence? This is hnRNA and this is mRNA. What are the sequences of bases transcribed from DNA and are found in both hnRNA and mRNA? So if you're asking about both hnRNA and mRNA, then this area that you see, it's an exon. This area that you see, it's also an exon. Over here, you will just have exons. So this is the thing that is common between hnRNA and mRNA. but this area which I have marked in yellow, it's intron and it is not found. This sequence is not found. You have to tell that intron is basically intervening sequence. Okay, intervening sequence which comes and messes up the whole system. Fine? Works? Okay? Will you be able to draw and make them understand that? It's of 2 marks, so you draw once and then make them understand this thing. Okay? And this process is known as splicing, as I discussed earlier. Cool? Next question. Given below are sequences of nucleotides in a particular mRNA and amino acids coded by it. So this is mRNA, children. Okay, write the properties of genetic code that can be correlated from the above given data. So first thing that I saw is it's AUG, and AUG is, I learned that AUG stands for methionine. Sorry, AUG is a start codon. AUG is a start codon. But over here I see AUG is coding for methionine. So I see two functions that I can see is basically start codon is also acting as the one that codes for methionine. Okay, now this, if you think it's not fine, then fine, chodo. If you see over here, UUU and UUC, both are coding for what? Phenylalanine. I see 2 codons for this. So I call this as degenerate. Okay, this character of genetic code is degenerate, as in it's a very degrading character. No, 2 amino acids are, 2 basically codons are coding for just one, one amino acid. Okay, so that is, this particular guy is thinking that I am coding for phenylalanine. This particular guy is also thinking that I am coding for phenylalanine, but they do not know that both of them are actually coding for phenylalanine. So this is a degenerate character of genetic code. So this is one character. The second. Next thing that I can see is what, children? UAA is not coded by anything. There is no amino acid that is coming. So basically I can understand that UAA probably is what? A stop codon. UAA is, children, a stop codon. Next thing that I can understand, do you see commas, anything, full stop, anything? No. So it is contiguous. The genetic code, so this is third one, and now the fourth one is that genetic code is contiguous. That means it is continuous in fashion. Works? Yeah? And do you see that GAG is coding for something else over here? GUG or GAG, is it coding for something else over here? GAG is for glutamic acid, GUG is for valine. Everything is same. Okay? So can I say it's unambiguous? It's not ambiguous. All the codons, there are no multiple codons for, sorry, I can say that codon is not coding for multiple amino acids. Codon is not coding for multiple amino acids. So that is something different which I usually didn't see over here. Okay, and this feature is known as unambiguous. The genetic code is unambiguous. Fine? Works. This is the 5th character that I see. Now, if you want the answer, you can write like this. The codon is triplet. Okay, yeah, each of them basically they are triplet codons. Okay, and 61 code for amino acid, 3 codons are for stop codons. Now, amino acids are coded by more than 1 codon, hence the code is degenerate. Okay, now the codon is read in mRNA contiguous fashion. There are no punctuations, we studied. The code is universal, okay, from bacteria too, but there are exceptions. You have mitochondrial codons and protozoans which are exceptions, okay. AUG is initiator codon and these are terminator codons, okay. And unambiguous is also there. I don't know why I'm not able to find over here. Anyways, so these are the things. And also triplet. Works? Do you get that sense of happiness while solving this question which I get over here? So again, a question for color blindness. So let's see what is the answer. So question number 17. In a family, the father, the daughter and the son are colorblind children. We have to make a note of this. What is this? Father is colorblind. So it will be XCY. Okay, this is the father. Now, the daughter and the son are also colorblind. So daughter should be XCC. Right? Because if there was a normal thing, then she would not have been colorblind, and son is also colorblind. Now what is happening, children? Whereas the mother has normal vision. Okay. The mother is said to have normal vision. Chalo. Okay. Do you think that the son and the daughter have inherited disease from their father? Okay, work out the cross to justify your answer. Chalo, let's start. Let's begin. So thinking that father, we already know that father is XCY. Okay, and it is claimed that the daughter, okay, the daughter that will be formed, at least one person will be having this condition, and the father, and the son which is being formed having XCY. Now, can I just blame the father for this condition? I don't think so. Why? Because, let's see, if this was normal, if the female was actually normal, then the case would have been kind of different. Let's see the case. So you have to show them. You have to draw this condition. This is condition number 1, case number 1. If the female was actually having normal vision, in that case, X is in the gamete, Y is in the gamete, X is in the gamete, another X is in the gamete. When they fuse, I get XXC. When they fuse, I get XX and C, right? If I fuse them, I get XY. If I fuse them, I get XY. The son would have been normal. The sons would have been normal. And the daughters would have been just carriers. And the daughters, 50% of the daughters which are there, would have been just the carriers. But do I see that here over here? No. Now case number 2. When the father obviously is colorblind, the mother is a carrier. In this case, let's see what's the condition. So over here, XC in one gamete, Y in another. X in one gamete and XC in another. Okay? This and this fuses, I get X, XC. This and this fuses, I get XC, XC. This and this fuses, I get XY. This and this fuses, I get XC, Y. Over here, I find son is colorblind, daughter is colorblind. Okay? Don't I see it over here also? So yes, 25% of daughters are carriers and 25% of son are normal. Okay, but this thing can I avoid? No. So this is the thing that is actually happening. So you have to draw and make them understand that this is a 3-marker question. You have to show case 1 and case 2 and make them understand things. Cool, children? Works? Can we go on to the next question? Yeah? Now, identify, this is 18th question. Identify the picture given below and the name of the tool under which it was viewed. What do you see this? What do you think this is? Worm? Some sort of worm? Is this that so? Well, this is bead on a string structure. Okay? And this was seen under electron microscope. The device that is being used is electron microscope. Now, what are the dark spots that represent over here? What do you think these dark spots are? So, dark spots that you see over here, children, are nothing but, so over here, 1, 2, 3, 4, 5, 6, 7, 8. You have 8 histone proteins. Okay, you call this as histone octamer. What do you call them? You call them histone octamer. Okay. Now this histone octamer is basically, what is this used for? So if you see the DNA, if you go around the DNA, if you calculate the number of everything, so basically 2.2 meters of our DNA is there. Okay, it's 2.2 meters. Can we accommodate that? No. Okay, if you want to accommodate that in a small nucleus, it's not possible. So for making them accommodable, what do? You coil them around something. Okay, you coil them around something. So basically the histone proteins on which the DNA wraps around. It's this particular histone proteins on which the DNA wraps around. And then it goes like this. Again, you will be having another histone protein on which again it will wrap around. And the same thing goes on. These are the beads that you see. So this whole structure, when you have the DNA plus the histone protein, what do you call this? You call this as nucleosome. What do you call this? You call this as nucleosome. Okay, and the whole structure of the DNA that is being formed over here, when they are twisted, they are coiled and everything, you call the whole thing as chromatin. You call the whole thing as, children, chromatin. Now chromatin, when it condenses more, when this chromatin, it condenses more, basically what it is forming, it will form the chromosome. So over here you will be finding beads, all of them, they will be condensing around here, and this will be reeling down to chromosome. That is the most condensed form. Okay, so this structure is known as nucleosome, which is the histone proteins plus the DNA, and there are 200 base pairs of DNA over here. 200 base pairs of DNA which are being coiled around over here. Okay, how they are formed? This is how they are formed, and why they are formed? Because you want to arrange this 2.2 meters of DNA together somewhere, right? And there should be some way to do that, and that is this. Then later on, this chromatin arranges themselves into chromosome during cell division. Works? Okay. Now one more thing that you should be knowing is that there is one more histone protein between the two strands. Which one? Okay, there is one more histone protein that is over here as well in between the two strands. So when they are super coiled, they are all together. They all look to be very close to each other. When they are not, they are loosely packed. Fine. So this is the structure that you need to know. Will you forget this? I hope you don't. Going ahead, 19th question. Explain the process by which amino acid gets attached to the tRNA molecule during translation process. So this is 1 mark, 1 mark, 1 mark. So just we need to put little of explanation. Okay. So what happens is that the tRNA okay, it has 5' end and then it has 3' end. On the 3' end, children, it attaches to an amino acid, and it's not just random amino acid that it attaches to. It also have an anticodon over here. So for example, U is there. This loop is known as anticodon loop. Okay, suppose this tRNA is there with this amino acid, that is phenylalanine. Okay, now it's there. This anticodon loop will go and attach to the codon. And how do you find the codon? Basically, over here, this is an mRNA. An mRNA has a sequence AAA. Suppose. So AAA sees tRNA, this anticodon comes and binds with this particular codon. And that's how you read it. So yeah, this found the perfect match and it will give out the amino acid. But the initial thing, how does the amino acid come there? This is also known as, amino acylation. Amino acylation, or you say charging of sugar or charging of tRNA, or you say charging of tRNA. And this thing, basically this particular process requires lots and lots of amount of energy, children. This is a high energy requirement process. Okay, you need ATP's energy, lots of energy for this charging of things to be done, amino acid to be done, right? So tRNA is the one that picks the amino acid and goes away like that, and goes and gives to this while reading the mRNA codon. Fine. Now how does translation process gets terminated? How does it get terminated? So it goes on. There will be different tRNA, but now comes a place where you are come across UAA. This is a codon. It's a stop codon. At this point of time, there won't be any tRNA. There won't be any tRNA, okay, which will be coming over here to bind and give an amino acid. So now it will be stopped. So tRNA came over here, it was fine, but there is no tRNA that will come over here, okay, and bind, because this does not have an anticodon. No anticodon. And hence this will be terminated. So is UAA the only stop codon? No, children. You have to remember 3 stop codons: UAA, UGA, UAG. They are all stop codons, and if stop codons are there, then the process of translation, that is, translation is what? Basically the process of forming protein from the mRNA. So over here mRNA is red and the amino acids are there, different amino acids will come together, they will form peptide bond between each other, and that's when you get amino acids coming together, you get what, children? You get protein. Fine. Now expand UTR and tell where are they located. Where do you think UTR is located? What do you mean by UTR? Expand UTR, it's very simple. UTR means untranslated regions. Untranslated regions. Okay. And where do you think UTR is located? UTR, basically, if I say that this is the normal thing, okay, translation region where things have to be translated, at one end, okay, in the fifth end and the 3' end, in the 3' end, near the 3' end and the 5' end, you find this area which will not get translated. This area which won't be getting translated. You call this as untranslated regions on both the sides. So you say 3' end and 5' end towards both the sides of this, you will be finding untranslated regions as well, which won't be undergoing translation. Cool? Works? Now going ahead. Stability as one of the properties of genetic material was very evident. This is the 5-marker question that we have entered, children. Okay, stability as one of the properties of genetic material was very evident in one of the very early experiments in genetics. Name the scientist and describe the experiment. State the conclusion he arrived at. So basically, genetic materials to be stable, it is a very important thing. Okay, so there was a scientist who was working on what is a genetic material, what could be the genetic material, RNA, DNA or a protein, and his name was Frederick Griffith. His name was Frederick, I hope I'm spelling is correct, okay, Griffith. He was working on an experiment where he used mice. I don't know why I was going to write rice because I'm hungry, I think. And also a bacteria that causes pneumonia, which is known as Streptococcus pneumoniae. It was known as pneumonococcus as well. Okay, and this bacteria had 2 strains, children. One strain was R strain and the other strain was S strain. The S strain had a coating around it, had a smooth coating around it. Okay, it was made up of polysaccharides. And this particular coating actually made this particular strain of bacteria virulent, as in it was causing the disease that is pneumonia. It was able to cause the disease that is pneumonia. Okay, whereas when I talk about R strain didn't have, it has a very rough surface, didn't have that extra layer. Okay, it was non-virulent. Fine. Now the things that he did was, first thing, he took the R strain, okay, live R strain, the living bacteria, he put it in the mice. He found that the mice was still living. Okay. He took the S strain, the one that had the smooth covering, okay. He put it in the mice and he found that the mice died. So he got to know that possibly something is getting passed on from these bacteria to this mice, which is making this mice kill itself, will die. Then what he did was, he did want to do much more experiment, little more. He heat killed the S strain. Okay, he treated with heat and he thought he killed something. Okay, now he treated with the mice and he found that the mice was alive. Okay, S strain, which was virulent, is now not showing the virulent character because it was heat killed. Okay, but then what he did was, he took the S strain, heat killed S strain, and then he mixed with R strain that was live. R strain usually didn't cause any problem, but now he has mixed them and now he has inserted and he found that the mice is dead. But the problem was, S strain heat killed was not killing the mice over here. How can it kill it there? How was it able to kill it there? Probably something in it was not dead. Something in it was very stable, Okay, something in this was very stable and it was passed from this guy to this guy. It was passed from this guy to this guy, making this R strain what, children? S strain. Making this R strain turn into S strain, transform into S strain. And this experiment also you call the transforming principle experiment. What do you call this experiment as? Transforming principle experiment. Okay, where he saw that S strain was giving something to R strain, and that something is getting making the R strain transform into S strain, which is why this particular mice is getting dead, just like how it was getting dead over here. What was that something, children? Even though he was not able to find it out, there were lots of other people who came into play, that is Avery, MacLeod, McCarthy, all of them came into play. MacLeod, McCarthy, all of them came into play. They said that DNA is a genetic material that is getting transformed, that is getting passed from this guy to this guy. But then some other person came. Okay, some other person came who finally said that no, not the other person. Basically, two people came, Hershey and Chase. They said that yes, DNA is the genetic material that is getting passed on. They are the ones who finally found it out. Okay, works. So this was the experiment, children, and you have to explain it in a very nice way. Just like how I said, in the written format, you can say, instead of making the mice, you can write live mice. You can write live mice over here, and you can write dead mice. Live mice, and you can write dead mice. Okay, over here you just put arrow, train, rather than putting injection and everything, just show like that and explain it to them point by point. Explain number 1. Case 1, this is the case. Case 2, this is the case. Case 3, this is the case. Case 4, just like that you have to explain. Okay, works? So he was able to understand that something is making things transform from one form to another. This is the genetic material probably. Okay, this is the genetic material and it is very stable. This genetic material has to be very stable. That is why it is not getting killed over here. Under heat also it was not getting killed. It is now getting transformed. It is now getting from here to here it went. So that is not heat killed. Genetic material was safe. Okay, works? Now going ahead. Let me just see the time. Yeah, okay. A tall pea plant bearing violet flowers. Okay. Tall pea plant. It can be tall like this. It can be tall like this. Two conditions. Okay. Bearing violet flowers. Violet flower is also a dominant character, children. Violet flower, I write it like this. Usually that's how genetics how you write. W stands for violet and small w stands for white. So even though white is recessive, you use capital W only. Okay. So violet flowers can be again like this or it can be like this. Okay. Now in this case it can be, the conditions can be like this, children. The condition can be like this, right? Or it can be like this also. So many conditions are there which makes the sense that it's what, tall and white. All of them basically are tall and white. Whatever you do, all of them are tall and white. Fine. Now, okay, one more condition. This is also tall and white. Okay, works? Okay, so a tall pea plant bearing violet flowers with unknown genotype is given. So you do not know what is the genotype. Now I have already told you, but still, okay. Now, find the genotype by working out different crosses only by selfing the plants. So you can do one thing only, that is selfing. Okay, you can do selfing and then you can try to find that what is the genotype. Write the genotypic and phenotypic ratio of each cross that is shown by you. So, to make your life easy, first thing is capital W, capital T, capital W, capital W. So over here, the plant is having capital T, capital W, visible? Okay, or I'll write it over here. This is one parent, this is another parent, children. Okay, I am assuming capital T on one chromosome, another chromosome capital T, capital W, capital W. Okay, so this chromosome can go with this. So anyways, it can be capital W, capital T, capital W, capital T, capital W, capital T, capital W. Whatever you do, you get this only. This is a set of chromosomes. This is the gametes that you get, whatever you do, from one parent and another parent. When you cross, all of them will be capital T, and capital D, capital W, capital W. So all of them, children, if you say the genotype is basically 100%, it's 1. Okay, phenotype is also like this. Phenotype is also 1%. Basically all of them 100% similar. Okay, they are 100% same. Now second condition, when the genotype is like this. In this case, there is capital T, capital W, small w. Now if I am selfing capital T, capital W, small w, right? So on the chromosome, I can have capital T, over here capital T, over here I can have capital W, and over here I have small w. So this and this can go, so it will be capital T, capital W, 1. Okay, this can go, so it will be capital T, small w. Similarly this, so same thing that you will get these two different types of, so this is capital 1. Okay, similarly here also you will get the same thing because of the same parent. Whatever you do, you get these two type of gametes. Now when you cross, when you put two of them there and two of them here and do the Punnett square, you find this is the thing that is being formed. Okay, so out of this, now you see some variation. So over here, I saw that this is capital T, capital W. That means this is tall and violet in color. This is what? Tall and violet in color. Now next over here also, this is tall and this is violet in color. This is also tall and violet in color, but this is tall and white in color. So now the phenotypic ratio is what, children? 3:1. Phenotypic, how does it look? So all of them are tall and violet, but this is tall and white. So the phenotypic ratio becomes 3:1. Okay, what about the genotypic ratio? What about the genotypic ratio over here, children? Genotypic ratio. Chalo. This, like this, I only see this one, so 1. Okay, like this I see 2, so 1:2. Like this I see only 1, so 1:2:1 is the genotypic ratio. Okay, the genetic makeup is kind of different. Fine, this is the second case. Next, going with this, here capital T, small t, and capital W is there. Over here capital T, small t, capital W. The chromosomes, you see, it's capital T or it can be small t. It's capital W anyways and it can be small w. Oh, sorry, capital W only. Okay. Now if I make the gamete, this and this can go together. This and this can also go together. Okay. So whatever I do, I get something like this always. This then this can go together, that is small t and capital W. Here also small t and capital W. So whatever combinations I do, I get these type of combinations. Two like this I will be getting, and two like this I will be getting. I will put down over here and I try to cross them. What do I find? This is tall and violet. This is also tall and violet. This is also tall and violet. This is what, children? Dwarf and violet. Again, 3 of them are tall and violet and one of them is different. It is dwarf and violet. That is a phenotypic ratio, 3:1. Now what about the genotypic ratio? This is one type of genotype that I see. This is both of them, basically I see 2 of them like that and one of them like this. So genotypic ratio, if I see, genotypic ratio becomes what? 1:2:1. Okay, 1:2:1. Over here, this is the case when you did with round and yellow. Okay, so here you get 9:3:1. I told you I will be explaining you this, right? So over here you see, over here the parent is, children, capital T, small t, capital W, small w. Okay, again none of the parent is capital T, small t, capital W, small w. The chromosomes, this is chromosome 1, chromosome 2. Okay, so this is capital T, this is small t, this is capital W, this is small w. This can go with this, so it will be capital T, capital W. This can go with this, so it will be capital T, small w. Okay, this can go with this, so I get small t, capital W. This can go with this, so I get small t, small w. I get 4 type of gametes, children. Over here I get 4 type of gametes. Similarly over there also I get 4 type of gametes. 4 gametes I put here, 4 gametes I put there. I cross them, I get these many things. Okay, and now what is the phenotypic ratio? 9:3:3:1. And can blindly say what will be 9, what will be 3 and what will be 3 and what is the 1. How can I say that? Listen, 9 you always attribute to the parent character. Okay, the dominant homozygous one. So it has to be, you think like that, okay? It will be tall and violet. No recombinations, okay? The dominant characters, tall and violet is the dominant character, you think like that. All of them will be tall and violet. One will be the recessive character, dwarf and white. Rest of them is the combination of both of them, okay? So 3 will be tall and violet is done, so it will be white. Next 3 will be dwarf and white is done, so this will be violet. So this is the phenotypic ratio, children. This is how they look, okay? This is 9:3:3:1. What about the genotypic ratio? What about the genotypic ratio over here? So genotypic ratio will be this, children. Okay, genotypic ratio here WV is mentioned, but understand it's W only, okay? So over here, how can I explain it to you? It is 1:, no one will ask you to tell the entire genotype. It is 1:2:1, 1:2:1, and in between just put 2:4:2. In between just put 2:4:2. Again I'll explain. Put 1:2:1, 1:2:1, then double it in between. 2:4:2. This one is the genotypic ratio for F2 generation of dihybrid cross, and this is the dihybrid cross. Okay, this was the one that explained independent assortment. So since tall was not always going with violet, which is why we got these type of features. Okay, so it was independently assorting all the genes. Works? So I hope we are done. In this case though, obviously everyone will be tall only. So we did this. How did I just repeat anything? Yeah, these things are same. Okay, so we have 4 things, and 4 things we have already completed. Works? So you have to understand that how it can be tall and violet. You have to make sure you know this, then only you will know the answer. Otherwise you would not. Tall and violet both are the dominant characters. If you are not aware of that, you won't be able to solve this answer. Okay, going ahead. Many patterns of inheritance deviate considerably from the pattern of inheritance as explained by Mendel. List any 4 such inheritance patterns and explain any 3 with the help of examples each. So Mendel had his own set of things. Okay, so one thing that he said is, if there is capital T and small t, capital T, if this is a dominant trait and this is a recessive trait, capital T will totally dominate and this will be tall. But in the case of Snapdragon, okay, in the case of Snapdragon, what was seen is that, I told you already, right? We had capital R, small r. So this was red, this was white. But when now the progenies were made, okay, what happened when I crossed them? All of them were capital R, small r, and all of them, rather than being red, they were pink. That means it was incompletely dominant. The capital trait was incompletely dominant over the small trait. Got it? So that is incomplete dominance. In this pattern, neither allele is completely dominant over the other, and the phenotype is a blend of both the alleles. In Snapdragons, crossing a red flower plants with white flower plants results in pink flowers, where both the alleles influence the flower color. Okay. Next is codominance. Okay. So according to him, again, they cannot be expressed together. Obviously, both of them cannot be expressed together. There is no middle height, nothing like that. But it was seen when you spoke about blood groups. When they spoke about blood groups, okay, so there are 3 blood groups, children. A, okay, should I make it somewhere else? Yeah. So there are 3 blood groups, A, B and O. Okay. This is explained by this is explained by gene B, and this is basically I gene, nothing. Okay. So when there are 2 copies of IA, this blood group is A. When there is IA and there is I, okay, this A is dominant over this, which is why again it will be A blood group only. Similarly over here for B, if it is IB from one parent, from other parent is IB, then it will be B. From one parent if it's IB and from other parents is I, then it will be B, because B is dominant over O again. So until now it was all fine. Similarly for I also, if there are 2 copies of I, it's a recessive trait, so that's when O comes. Okay, but when from one parent you get A and from other parent you get B, the blood group was AB. A was not fully dominant over B, nor B was fully dominant over D, which is why You call this as codominance. Codominance. This was also not taught by Mendel, but it came to action. So this is an example. And also Mendel said that there were limited alleles. There were limited alleles. So for height, tall, this khatam. But over here we see 3 alleles are there. There are multiple alleles that are being shown. That is also one more thing. So codominance, where both the alleles in a heterozygous individual are fully heterozygous, as in they are not same. A and B are different, heterozygous. A and A, it is homozygous. So both the alleles in heterozygous individual are fully expressed, leading to a phenotype that shows both the traits equally. Example, in human beings, ABO blood group show codominance. A person with one allele for type A blood group and one allele for type B blood group will have AB blood type, where both A and B antigens are equally expressed. Okay. So on this, they have the antigens which are being expressed. Now multiple alleles is also one such example. You are asked for 4 things. You are asked for list any 4 such characters. So fourth one is polygeny. Polygeny is when you have already learned that basically there is one gene, there is only one gene that will be, gene have different patterns, there will be one. There is one allele for tall, one allele for this thing. But this is one gene for height. But over here in polygeny, it means there are multiple genes, and these multiple genes are responsible for what, children? The skin color. So combined effect of all these genes will decide the skin color. It's not decided by one gene. So this is an example of polygeny, where multiple genes are coming together to give the skin color. There is pleiotropy as well, given in your NCERT. What do you mean by pleiotropy? Where basically what happens? Suppose mutation in one gene is mutation in one gene. Is going to affect 2 character. This is one character, this is the second character. So basically that means one gene is responsible for both the characters. Okay. So this is known as pleiotropy. Pleiotropy and polygeny, please do not get confused. Polygeny is where you have multiple genes for one character, that is skin color. Okay. Pleiotropy is where you have one gene for multiple characters. Fine. Done? 22nd done? Can we go ahead? 23rd. Construct a transcription unit with a coding strand given below with proper labeling. Okay, we have to find the coding strand. This is the coding strand. We have to make a transcription unit. So it is A, T, G, C, T, G, A, T, G, C, G, A. Right? This is the coding strand. So the template strand, the transcription unit is a template strand. So over here, okay, so DNA only. So it should be T, it will be A, it will be C, G, A, C, T, A, C, G, C, T. This is the transcription unit, children. And over here you will be having the promoter region. Okay, there will be promoter region, and there will be, this is the structural gene, and there will be a terminator region as well. Okay, there will be a terminator region, there will be promoter. You studied this, right? Where did that go? Oh yeah, like this. Okay, there will be a promoter region, and there will be a terminator region as well. No. Okay, we were on which question? Okay, so here also there will be codes. Here also there will be codes. Fine, works. When does a coding strand become a template strand? Never. A coding strand will never become a template strand. First thing, how can it become a template strand? Because obviously this one is also already there. It's a template strand. Okay, so it will never become a template strand. But if I say I want to make a protein out of it, okay, at this point of time, mRNA comes and dashing to that area and going to this in that direction, it will become a template, that will become a coding. But that's right, basically we generally do not say the coding strand can become a template strand. Okay. Now, why does a double helix DNA molecule transcribe into single stranded RNA molecule? Because double DNA is transcribing into a single RNA strand. Why is that so? Because only one strand is being copied. Children, only one strand is being transcribed, and that is the template strand. Do you expect that coding strand is also getting transcribed? No, it's only the template strand that is getting transcribed, which is why, okay, template strand is only getting transcribed, which is why you get single stranded RNA. If both of them are transcribed, what will happen? You will get double stranded RNA. You will get double stranded RNA. Is it okay for you? This double stranded RNA, do you think it can undergo translation? Do you think it can undergo translation? No, children. Also, this is set for making a protein. This is another type of protein that will be made from this. It will be lot of confusion within the cell itself. Okay, so we do not want all of that, which is why there is only one helix that is being read. Okay, or one single stranded RNA that is being formed. Cool. Going ahead, draw a schematic self-explanatory label diagram of Lac-Operon in switched on condition. It's a very interesting question. So Lac-Operon is my favorite. So Lac-Operon, if I say this is a Lac-Operon, my dear children, okay, this is a promoter of one gene, this is inhibitory gene, this is promoter of our gene, this is the operator, this is the structural part of the gene, Z, Y and A. Okay, fine, and there will be the terminator as well. That is the terminator portion, so I am not writing it there. So children, understand that this is a gene. This thing that you see is a gene, is a structural gene that is known as regulatory gene. Okay, and regulatory gene is the one that codes for repressor protein. This is inhibitory regulatory gene. Okay, this has inhibitory effect. So this is repressor protein that it makes. There is also one more thing that makes activator proteins, but here repressor it has, and this is the promoter for this particular gene. Okay, so RNA polymerase comes, binds to it, and then transcribes it. So promoter is that region where RNA polymerase comes, bind to it. Now this is our next set of gene that is present over here. This is a structural gene that I have over here. Okay, that is a structural gene that I have over here. Okay, this is the operator of that gene, and this is the promoter of this particular gene. Now over here, Z stands for beta galactosidase, Y stands for permease, and A stands for trans-acetylase. What does that mean? Basically, this is all happening in the E. coli. This is E. coli's thing. Okay. Now, E. coli, suppose it is surrounded by a medium that is rich in lactose. That is rich in what, children? It is rich in lactose. Now, E. coli wants this. E. coli basically wants this. What happens is that E. coli take the lactose with the help of permease. So permease is the enzyme that will break down the membrane and such that the lactose can get inside. Such that, children, the lactose basically can get inside. Okay, beta galactosidase will later break down the lactose into what? Glucose and galactosidase. And children, galactosylate. Trans-acetylase will basically, there are so many unwanted things that is being found, toxic things, it makes it less toxic. Okay, so usually what happens is that this repressor protein goes and binds on the operator, okay, and does not allow this transcription to take place. But when lactose or allolactose is there, they will act as inducers. Okay, they will act as inducers. They go bind to the repressor such that there is no one to bind over here and it's already active. This is switched on condition. This is switched on condition where there is nothing on the operator and everything is working really fine. And why do I call the regulation of Lac-Operon is referred to as negative regulation? Why, children? Because over here always there is repressor protein that is being bound, and this negatively regulates this negatively regulates the lac operon. It does not allow transcription to take place, which is why it is known as negative regulation. So in the open condition, you don't have to put any repressor protein over here, just like how I made it earlier, you have to make it. Cool? Works? Can we go ahead? 25th question, the last question. Identify the polarity of X and Y in the above given diagram, and how many more amino acids are expected to be added to this elongated polypeptide chain. So this chain should be in 5' to 3' direction. This mRNA, X is 5' and Y is 3' children. Okay? And how many more amino acids are expected to be added to this elongated chain? You see there's UAA, and UAA stands for what, children? Stop codon. Can I expect more amino acids to be added? No, stop codon does not have amino acids. So I cannot expect any amino acids to be added to there. Okay? Now, which sequence of bases are generally found at the X and the Y polarity? What type of sequences are found in X and the Y polarity generally? So X polarity will be having AUG, which is the initiator codon. Initiator codon. And AUG also codes for what, children? Methionine. Okay? Whereas this Y codon will always have UAA or UGA or UAG. They are all the stop codons that you can find, so that the translation process, it can stop. Okay? Mention the anticodon for alanine and valine amino acid based on the diagram. So alanine and valine, this is alanine and this is valine. Okay, if we cannot see, alanine over here in the case of mRNA, it is written GCA. You have to find alanine's anticodon. What will be the anticodon of alanine? If it's G here, it will be C there, C here, so it will be G there, A here, so it will be U there. Okay, and valine, if you see, it's GUU. So G there, C, U, I mean A, U means A. So over here, this is for valine. This is the anticodon for valine. Cool. Now, what is the composition of the catalyst involved in the peptide bond formation? So here, the peptide bonds are formed between all the amino acid. So who makes this? What is the composition? Basically, it's just an RNA. It's just an RNA, children. It's just an RNA, and this RNA is known as rRNA, ribosomal RNA, which is also acting as a structural RNA. It makes the ribosome. It is making the ribosome, and I have a specific thing. In the case of bacteria, it is 23S rRNA, sorry, 23S rRNA, I'll write properly. Understand, it is rRNA that forms the structure. Along with rRNA, you have ribosomal proteins. Okay, so this is 23S rRNA that makes it. And after that, apart from bacteria, if you talk about human beings, we have 28S rRNA that is acting as enzyme. What do you call them? You call them ribozyme, children. So this type of RNA, you call them ribozyme, and those are the ones which basically, okay, which basically helps in the peptide bond formation over here. Works? So that's all, my dear little champions. That's all for today. I hope that you understood each and every part that I tried to explain you. If you think that there is some confusion still now, I am here. You can comment it down. I'll surely try from my end to solve all your doubts, because that matters to me. Yeah. And if you like the video, then definitely do not forget to comment down. Tell me it was a good. I'll also be encouraged. Yeah. And I wish you all the best for your midterm. Everything is going to be really, really good. Do not be worried about anything. Okay. And do not forget to subscribe to our channel, and have a really, really good day. Bye-bye, dear. God bless you all.